joining us for today's seminar, which is part of the monthly Monash Micro-Imaging Advanced Microscopy seminar series. My name is Natasha, your host for today. I'm a Senior Research Officer at the Ramashiri Center for Cryo-EM at Monash. Before beginning, I would like to acknowledge the traditional owners of the land. I'm coming to you today from the lands of the Wurundjeri and Bunurong people of the Kulin Nations. I would like to invite everyone to pay respect to the traditional custodians of the lands and waters on which we stand. It is my great pleasure to introduce our guest speaker, Associate Professor Stefanie Redemann. Stefanie obtained her PhD in 2009 in the labs of Professors Tony Hyman and Jonathan Howard, Max Planck Institute of Cell Biology and Genetics at Dresden. She investigated the role of the actomyosin cortex in force generation and spindle positioning in the nematode C. elegans. This gave her the opportunity to combine her passion for cell biology and physics. Later, Stefanie joined the lab of Professor Thomas Müller-Reichert at the Medical Theoretical Center, Dresden, where she worked on reconstructing the mitotic spindle using 3D electron tomography. Since 2018, Stefanie has led her own research group at the University of Virginia School of Medicine, where she's currently using interdisciplinary approaches to study spindle assembly and chromosomal segregation in mitosis and meiosis. She has made significant contributions to our understanding of mitotic spindle architecture and microtubule dynamics during these cellular events. And she was also recently featured in the Journal of Cell Science as a scientist to watch. So today gives us the opportunity to be learning more about her exciting work, in particular, how she has used advanced microscopy cryo-EM techniques to better understand the architecture of spindle microtubules and the function of associated motor proteins in mitosis. I would like to now introduce Stefanie's talk titled High Resolution Imaging of Spindle Structure in Mitosis from Plastic to Cryo. Please enjoy the seminar. Stefanie, all yours.
Yeah, thank you, Natasha, for the very kind introduction. And I guess good morning, everyone, at this point, even so it's 7 p.m. over here in Virginia. So as Natasha said, I'm Stefanie Redemann, and I'm an associate professor at the University of Virginia at School of Medicine in the Department of Molecular Physiology and Biophysics, as well as cell biology. And so today I'm going to talk about microtubule organization within the metaphase spindle from plastic to cryo-electron tomography. Here we go. Okay, and so just broadly to introduce my lab. So what I'm really interested in is how spindles in mitosis and meiosis assemble and how those spindles can segregate chromosomes. And so if you think about spindles, they're made of many very small nanoscale components, right? You have microtubules, you can see here in green. You have motor proteins and chromosomes, and all of those kind of magically come together and form a bipolar spindle that then can generate sufficient forces to actually segregate the chromosomes during cell division. And so we really want to understand how these nanoscale components that are also intrinsically highly dynamic, right? So microtubules turn over within about 10 seconds, even so spindles are there for minutes and hours, and motors kind of shuttle back and forth permanently. So how can those really small, highly dynamic building blocks actually assemble a stable micron scale structure, and how can they then work together to generate sufficient forces to segregate the chromosomes? And so if you look at the classic textbook model of a spindle, you can see this here, right? This is taken from the Allbirds, one of the classic cell biology books. You can see that a spindle looks like this. There's microtubules, they come from a spindle pole, and some of those microtubules connect to the kinetochores. And here is the light microscopy picture that is at the basis of this cartoon. However, I find if you really want to understand how the spindle structure truly looks beyond this cartoon, then I would say that the microtubules in this light microscopy image are really not resolved. And it's really hard to make a statement on whether this cartoon is correct or incorrect. And so what we really need, if you want to understand spindle mechanics, is a better resolution. So instead of having something like this that you get by light microscopy, you want maybe something like this, right? Where you can see every individual microtubule, every building block, and you can start being very quantitative, right? You could count them. you could measure their lengths and everything else. And so I want to start with just introducing how we actually get to an image like this of the spindle, and also something that has been for a long time the core of the lab. And so what we generally do in the lab is we high pressure freeze our samples. So in earlier cases, for example, C. elegans embryos, but it could be tissue culture cells. And so what we do is we do high pressure freezing, which really just means that the samples are very rapidly frozen under high pressure, so about 2000 bar with 20,000 Kelvin per second, to avoid formation of ice crystals, right? Which would be detrimental for the ultrastructure. And here you can just see a comparison. So this example is Monash, and you can have conventional chemical fixation for electron microscopy. And then if you look at the cryofixation, you can see that the structural preservation is much better. And so, as I said, we use Leica ice. We do high pressure freezing of our samples. And for most of the plastic EM that we do, these samples are free substituted. So that means that at very low temperature, we very slowly remove the water and add fixatives. So for example, osmium and glutaraldehyde. We do then an EPON embedding. So EPON is a plastic resin, and ultimately do serial sectioning of our sample. And so what you see here is a little grid. And we really try to collect every single section of our sample. So if this, for example, was a cell, we really cut a lot of individual 200 nanometer thick sections to collect the entire sample. We then take it to the electron microscope and do electron tomography of these 200 nanometer thick sections. And this allows us to obtain a larger 3D volume. And for those that are not familiar, what we do in the tomography is that we put the sample in the electron beam, and then we rotate the sample in 1 degree steps. And at each degree tilt, we take an image. And so we end up with a whole set of images from different tilt orientations, so up to mostly 60 and minus 60. And those images can then be back projected into a 3-dimensional volume. Because we want to look at mitotic spindles, and those are not very small objects, we actually have to acquire a larger number of tomograms. And so what you can see here, that using our setup here that has a 4x4 camera, and at the resolution we usually work at, which is 2000 5000x, so 2.3 nanometer pixel size, we have to acquire 2 tomograms in order to cover the entire area. And so you can see there's 1 tomogram. The chromosomes are located here. Spindle poles would be there. This is the other tomogram. Again, here are the chromosomes, same as here. And there's the spindle pole. And each of these tomograms is 10 by 5 microns. And so we acquire these 2 individual tomograms on our Techni F20 using CRLAM. And we acquire many of those tomograms because we also want to cover a 3-dimensional structure. So each of the individual sections we cut, each of those that contain a spindle image, we will image. And so you can see that we have a whole set of individual tomograms also in Z direction. And so, for example, in C. elegans, this would be about 25. We're also currently working on mouse spindles, and there it's more in the range of 50 consecutive sections. Okay, so once we have acquired these 2 individual tomograms, we can use iMod or 3D mod from Boulder to stitch those in X and Y based on this little overlap area that you see here, right? And so this now gives us one section in X and Y that contains the entire spindle material. Okay, so the next step that we have to do is the segmentation of microtubules in these data sets, because these are the main building blocks of spindles. And we have initially used Amira, which now belongs to FEI and Thermo Fisher. And we're using a custom-made software that uses a template cylinder. And this template cylinder, you can see this here, has the diameters of a microtubule. And it looks in every voxel of our tomogram whether it can find a microtubule. And then it looks in the neighboring voxels and it generates a so-called correlation field. And this allows us, you can see this here, the segmentation of microtubules in our tomographic volumes. The calculation time is about 5 hours per tomogram. The kind of drawback of this is that it requires a significant amount of manual correction, right? Because it uses this template cylinder. It can also find mitochondria, for example, because they have a double membrane and other structures. And so we basically go in after the segmentation and manually correct every microtubule that the software has found. And this is or has been our bottleneck so far, because this can really take several days, depending on the quality of the tomogram and also the amount of microtubules. So some of these sections have thousands of microtubules, so you can envision that correction takes rather long. We recently, together with the Müller-Reichert lab and Robert Kiewicz, who is here, have developed a different software. It's called TARDIS, or Transformer-Based Rapid Dimensionless Instant Segmentation. And this is freely available. So if you're interested, you can check it out here. And this software does a much better job in actually detecting microtubules. And it can do this in TEM images, but actually also in cryo-ET. And we've also set it up, at least for cryo-ET, that it can detect membranes. And so if you compare this to a mirror, looking in these images, or also in the quantification, this software is much faster and also has a much higher accuracy. And so we are basically much, much faster in now segmenting all the microtubules in our individual tomograms. So at this point, it doesn't take several days, but often can be achieved in an afternoon. And you can envision that this really is a big game changer. Okay, so once we have segmented the microtubules in every individual tomogram, we actually use a mirror again to stitch these individual tomograms in Z. So you can see this here. These little blocks are tomograms, and these lines are just to highlight microtubules that are stitched across the Z direction now. And so this really enables us to get a complete three-dimensional reconstruction of a spindle. We can also add other features. So for example, here we've added the nuclear envelope and chromosomes, and this has been done manually, but there's also now new machine learning tools from Arrivis. So some may know this software from the light microscopy. And so this really allows you to train the system, and you can segment membranes and mitochondria and chromosomes much easier. And then once we have stitched everything together, we can also do an annotation of different subgroups of microtubules, and I will talk about this soon. And we can run certain quantifications. So we have a pipeline in a mirror that allows us to quantify these reconstructions. And so this is just to show. So this is a metaphase spindle of the C. elegans embryo of the first cell. And you can see these are all the microtubules. Chromosomes are here in blue, microtubules in green, and microtubules that contact the chromosomes, kinetochore microtubules, are shown in orange. And so now you can envision, right? We can now see every single microtubules, and at this point we can start being really quantitative with the data and really try to understand how those spindles can maybe assemble. Okay, so the first thing we thought one could do with this data set is just try to classify microtubules, right? So here's again this cartoon of a spindle, and you can see that these microtubules can actually be grouped into different classes, right? You have kinetochore microtubules that attach to the chromosomes. You have spindle microtubules that are coming from the spindle poles, but do not attach to chromosomes, but grow towards the bulk of microtubules. And then you have astromicrotubules that grow out from the centrosomes and actually can interact with the cortex of the cell. And so we can take the reconstructed data and actually do exactly the same. And so sounds easier sometimes than it is, because if you look at these spindles, there's about 20,000 microtubules. And it's maybe easy to identify kinetochore microtubules based on their interaction of the chromosomes, but to distinguish between astromicrotubules and spindle microtubules is already not so straightforward. And so you have to come up with some geometric parameters that really allow you to kind of group these microtubules into different subclasses, right? And so we have done this here. So for example, we have identified kinetochore microtubules, and mainly based on their distance to chromosomes, because we can also, this is plastic data, right? We cannot see a kinetochore per se. They're not visible to us. We can only see the chromosomes, but you can envision that a kinetochore microtubule has to be close enough to a chromosome to actually be interacting. And so we chose a distance of 200 nanometers. And so you can select every microtubule that comes within this distance and refer to it as a kinetochore microtubules. And for spindle and aster microtubules, you can define an opening angle here that encompasses the metaphase microtubules, and every microtubule that is within this cone will be a spindle microtubules, and others will be aster microtubules. And so you can see that a complete spindle has about 20,000 microtubules, but only 500 of those actually make contact to the chromosomes, so a rather small number. The other question when we got this data was like, what other properties could we actually measure, right? Because no one had any type of this data. And so while, you have this reconstruction and it looks really pretty, you also kind of have to deduce some data from it and really make sense of it. And so after we classified the microtubules, we thought the other thing we could look at is microtubule length, right? And so if you are familiar with microtubules, right, there are these polymers that are assembled of tubulin dimers. And in their life, microtubules can grow. And ultimately, they undergo something that's called catastrophe, and they start to shrink. And so you can see this here. Again, this is a not playing Xenopus aster, but you could see, and I can maybe just play it again, or not, how microtubules grow out from the centrosome. And so if you would plot the length distribution of these microtubules, it would be exponential. And this is just because the longer the microtubules are, the higher is probability that they collapse. And I think of this often like a Jenga tower, right? The higher the tower is, the more likely it is to collapse. And so if you would plot the lengths of these towers, you would always end up with a lot of short ones and not so many long ones, right? And so you could use the length distribution of the microtubules in the spindle as a readout for microtubule dynamics. And so if we do this, you can see those are aster microtubules, and they have an exponential length distribution. There's spindle microtubules, and it's somewhat exponential. And then if you look at kinetochore microtubules, it looks very different, right? It looks very messy, but it's more or less a linear length distribution. Okay, and so what this already tells us, if you look at this length distribution, that if this picture here from the textbook would be correct, and if every kinetochore microtubule would start at the centrosome and reach the chromosomes, then the length distribution should have looked like this, right? They should all be more or less the same length, which is the distance from here to here. However, you can see that here, this is absolutely not true. So this already suggested that there's something off with this textbook image. And so what we found back then is that kinetochore microtubules actually do not connect to the spindle poles. You can see this here again in this cartoon. And you can also see this when you look at the localization of the kinetochore microtubule ends. So this is the spindle axis, this is the pole, this would be the chromosomes, and this is the number of ends or fraction of ends we have found in this distance. You can see that there's actually ends everywhere. Whereas if you look at spindle microtubules, or those that are not kinetochore microtubules, you can see that most of the ends fall within this 2 micrometer area. So really saying that, unlike the textbook model, shows kinetochore microtubules actually do not connect to spindle poles. Okay, and so this we found initially in C. elegans, but the Thomas Müller-Reichert lab also reconstructed HeLa cells, and you can see that in HeLa cells is very much the same. So you have kinetochore microtubules that are attaching to the chromosomes, and then you have non-kinetochore microtubules, and what they find is that those kinetochore microtubules also do not directly connect to the spindle poles, right? And so saying that this initial textbook model is somewhat incorrect, and if you wanted to modify it, it would look something like this, right? So showing that kinetochore microtubules are not directly connected to the pole. Okay. So next, we took a closer look at these spindle microtubules, and what we realized is that if you look at the length distribution, you can see that the average length of these microtubules is really short. So it's about 2 micrometer, even so the entire spindle is about 10, right? So you can see this here. And this really says that actually these microtubules do not span the entire spindle. So they are also not coming from the spindle pole and connecting to the chromosomes. Rather, the spindle is kind of an array of a lot of shorter microtubules that build this long spindle, right? Very different from the textbook image. And Just to say, this is true for any spindle we've looked so far. So you can see there is a meiotic spindles in C. elegans. This is the mitotic spindle in C. elegans, and this is actually half of a mouse spindle. And in all of these spindles, if you plot the length distribution, you can see that on average, microtubules are actually short, and they only span about 20% of the distance between microtube poles and chromosomes. So really saying that spindles are composed of not long continuous microtubules, but rather many short microtubules. Okay. The other thing we realized is that if you look at the textbook, you have microtubules that are connecting to the chromosomes, and then you have microtubules that come from the spindle poles, right? Like spindle and aster microtubules. But we discovered that if you look at this HeLa cell data reconstruction, there are a lot of microtubules that are neither nor. They're not connecting to the spindle pole, and they're also not connecting to chromosomes. You can see this here, and it's actually half of the microtubules, right? And so those are, let's say, undefined microtubules that have not been described before. And so they can really not be assigned to any of these classic microtubule subgroups. And so that was really interesting, and we really wanted to understand what are these undefined microtubules? What are they doing? Where are they coming from? What is their role in these spindles? Right, and just to update the cartoon, right, I already said, so kinetochore microtubules are not really connecting to the pole as they are here. And also not every microtubule is coming from the spindle pole, right? So there is a large fraction of microtubules that are short and that are not attaching to chromosomes or a spindle pole. So in order to learn more about these microtubules, we decided that maybe we needed yet a better resolution and maybe more insight to understand what those microtubules could be. And I think our initial hope was that we could maybe see some motor proteins or anything connecting to these microtubules. And so we decided to maybe move away from serial section plastic microscopy and try cryo-electron microscopy. And so just to put this in context, right? So before we did these large scale reconstructions, and they're kind of great because they provide a full structure and cellular context, but they come at a somewhat lower resolution of 2.3 nanometer. If we were going to do cryotomography, then we may get a higher resolution. But the problem is that because we're going to do this in a cell, we are really limited to this really small area that will be left for imaging. Because if you image in a cell, right, the way you have to do it is you have to use a FIPSEM. And so here we can use an aquilus to mill away everything you're not interested in and really just leave this 200 nanometer thick tiny lamella that then you can do cryo-electron tomography on. But the premise is that maybe we can achieve higher resolution and we can actually learn something new about those spindles that we cannot see in plastic. And so a lot of this work was done by a super talented postdoc, Will Conway, who's at the New York Center for Structural Biology. And so he really did champion all the cryo-electron tomography I will show you, right? And so what we did is we used RPE cells in this case. And because we are interested in mitosis, we actually first needed to enrich for mitotic cells because only about 2% of these RPE cells are actually in mitosis at any given time. So you can see here, right? But if you want to do cryo-ET, this is a really low number. And so the first thing we had to do is do something that's called a mitotic shake-off to actually enrich our sample for mitotic cells. And then we, and I'm going to skip a little bit of sample preparation. We decided to plunge freeze our sample. We tried different methods. We tried the waffle method and others. But at the end of the day, we decided to use plunge freezing of the sample. And so what we do is we use glycerol, 5% as a cryoprotectant and a very low blotting time. And you can see here, this is a frozen cell, right? And these are the lamellas we can produce. So this is the side view. And then here you can see the top view. Okay, and so just to sum this workflow up, right? So here you can see Will again in this reflection, and he's doing, he's growing the RPE1 cells. He's doing a mitotic shake-off to enrich for mitotic cells. We plunge freeze, and you can see those are the cells on the grid, right? And this is a zoom in. And we use a array scan with a liquid nitrogen stage to actually image those cells. And so you can see here is a tubulin as a marker and kinetochore, and you can really now see what stage the cell is in and whether you should or should not process it. We then do cryo-imaging, so you can see the cartoon here, right, where you basically mill everything away apart from a lamella that contains your region of interest. And here's how this looks on a grid. So this is a grid with several lamella, and this is again a lamella with an RPE cell. Okay, and so just to say, this is another picture, and what we did at the very end is also after we acquired tomograms of this data, we actually went and fit them all back into the lamellas in order to later be able to have somewhat of a context, right? To be able to know whether, for example, a spindle pole is located at the top here and the chromosomes were here, and which region of the spindle we are actually looking at, right? Because we now have a very small field of view. Nevertheless, it's really important for us to know how is the spindle oriented and where is the tomogram in respect to the overall spindle architecture. Okay, so this is how one of these tomograms looks, and all the acquisition parameters and processing are listed on the side. And so you can see maybe already some microtubules here. And so this is somewhere in the spindle. I'm going to help a little bit. So here we have annotated microtubules in green. There's a lot of ribosomes in these spindles and some membrane, which could be ER or maybe leftover of the nuclear envelope. And here you can see how this whole looks, right? So this is now a 200 nanometer thick tomogram of an RPE spindle in mitosis. Okay, so this is kind of cool, but of course, this is somewhat similar to what we could get in plastic, and our aim was really to get some additional information that we did not have yet. And so the first thing we looked at was just the neighbor density of microtubules, right? And I guess this we could have done in plastic too, but as you may or may not know, all the sample processing causes some significant amount of shrinking. And so if you do this in cryo, you can actually get the real distance between microtubules. And what we can see is that these microtubules have a spacing of 39 nanometer, and it's very characteristic. And so this really tells us that those spindles are kind of, the microtubules in the spindle are kind of behaving like pneumatics, like liquid crystals. But this is not maybe overwhelming. So we decided, okay, what else could we do? And so the next step is, of course, to subtomogram averaging. You can do this for the microtubules along at each helical repeat. And you can, for example, determine the number of protofilaments, which in our spindles was 13. You can compare this with in vitro assembled microtubules, which in this case are, for example, GMP CPC, so stabilized microtubules. And what we can see is also that if you, analyze the twist of microtubules, you can see that the GMP CPC microtubules are much more twisted than the microtubules in the spindle, right? Suggesting that maybe microtubules in the spindle might be more relaxed and less stable. Okay, but again, this the twist is somewhat interesting, but it's not something super exciting that we were hoping to get out of this data. And just to put a spoiler alert, as I said at the beginning, we were hoping to see any motors or microtubule end binding proteins. We have sadly not succeeded in this, and I guess this is just because averaging that would be required is really hard, contrast is really low, but we were actually able to measure polarity of microtubules in those samples, and this is something that we cannot get out of the plastic data. And so if you can see these images here, and I apologize now because they're in red and green, but what we find is that if you look at microtubules inside the spindles, we always see a high fraction of actually antiparallel microtubules, meaning that some microtubules have their plus end oriented towards the centrosome and others have it oriented towards the chromosomes. And we can determine this based on the orientation. You can see this here of these protofilaments in our tomographic reconstructions. And so this was interesting and this was new. And you can see the quantification here. So we find that there is like about 50% of antiparallel microtubules in these reconstructions. And so we were really curious where those microtubules come from, right? And so very naively, you could just think, okay, we have these spindles, right? And maybe we can determine the polarity of microtubules by just assigning the polarity in reference to the nearest pole. So let's say the microtubule end is here, and then it goes this way, then it will be closest to this pole. So therefore, the plus end will be here and the minus end will be here. If the end of a microtubule is closer to that pole, it would have the plus end here, the minus end here, and the plus end there, right? So you could just assign it based on the distance to the nearest pole, or you could assign it randomly. And of course, we have to have some way to verify this model and also the data we get of the electron microscope. And so one way to look at it is to use something that's called second harmonic generation microscopy. And so we do this together with two-photon microscopy. And so what you see here is a two-photon image of GFP tubulin in a mitotic spindle. And you can see here the second harmonic generation image. And so second harmonic generation allows you to determine polarity of filaments, right? And so if two microtubules are parallel, meaning they have their plus end at the same side, the signal will amplify and we will see a bright signal. And if they're antiparallel, the signal will be canceled and it will become dark. And if you have an image of overall tubulin in the spindle and the second harmonic, you can use this to calculate a polarity profile along the spindle. And so if you do this here and you plot the polarity profile, you can see that the second harmonic image gives you this type of polarity profile. If you now take a model in which we assign the polarity of microtubules to the closest pole, the polarity profile would look something like this. And if we assume that the polarity is random, it would look much more closer to this second harmonic image, suggesting really that the polarity of microtubules is really not spatially dependent. So it's independent of where it is located in the spindle. Okay, so next we were wondering, okay, maybe we're just looking at microtubules that are crossing over from the other side, right? A spindle is bipolar, and so you could just have microtubules from this pole crossing over at the other side, and that should give us a large fraction of antiparallel microtubules. And to test this, we made monopolar spindles, and you can see this here. And you can just do this by preventing the duplication of the spindle pole. And what we find is that those spindles still have antiparallel microtubules, right? So if you look at this, this is the quantification, this is the control, this is monopolar spindles. And again, those microtubules are still antiparallel. And if you put this in a cartoon, it would look something like this, right? You have a half of a spindle only, and microtubules have random polarity in these spindles. So you really do not need bipolarity, and we're not looking at microtubules that are coming in from the other side. Okay, so at this point, we were wondering, okay, maybe motor proteins could play a role in this process. And just very briefly, right, we know that spindle assembly is mainly driven by interactions between microtubules and motors, right? And so if you have microtubules, you have these motors, they can walk along microtubules, they can cross-link microtubules, and they can also slide microtubules in respect to each other, for example, sliding those microtubules away from each other. And so we really know that these interactions are driving spindle assembly. And so the most well-described motors here are actually kinesin 5 or EC5, which is a plus end directed motor, and dynein, which is a minus end directed motors. And so if you inhibit kinesin 5 in spindles, you actually also end up with monopolar spindles, right? You can see this here in the cartoon or here where you have a spindle pole, and the chromosomes are actually kind of radially oriented around this spindle. If you inhibit dynein, you can see now that this spindle is very misaligned and very messy, and those poles are not very nicely focused. And so we really know that these two motors are very important to shape the spindle in mitosis. And so what we decided is to investigate whether these motors play a role in microtubule polarity. And so what you can do is you can treat cells with an inhibitor of kinesin 5, monastrol in this case, and this causes monopolar spindles. However, I would call them somewhat fake monopolar spindles because these spindles do have two spindle poles that are just collapsed into one bigger pole, right? So there's still two centrosomes that are kind of collapsed together, and the chromosomes are pushed away from these spindle poles. And so interestingly, if we do cryo-ET on those spindles, you can now see that those are mainly composed of parallel microtubules, right? You see this here in the tomogram. There's one little outlier down here. And you can see the quantification here and a little cartoon. So really it's showing that if we inhibit this kinesin, it actually changes the parallelity of these microtubules, and we lose this fraction of antiparallel microtubules. We did the same for dynein. And so this is just a quantification because it's not super interesting, but what you can see here, this is wild type and this is dynein. So if you get rid of dynein, there was no change. So we maintain this antiparallel fraction. But what does change is the spacing of microtubules. And so you can see this here, right? At the beginning, I said that the neighbor spacing is about 39 nanometers. So you can see this here. If we deplete the kinesin, microtubules collapse. So the spacing is reduced. It's only 31 nanometers. And if we deplete dynein, you can see that there's a variation in the spacing. You see there's multiple peaks. So actually the lattice spacing expands, and you can, I think, also see that this probably causes this severe misorganization. I think what is also interesting and what is something we're working on right now is if actually in spindles you knock out dynein, right, you get this very misaligned spindles. You can see this here, and this is data from Sophie Dumont's lab. But if on top of this you inhibit kinesin 5, you can actually restore bipolarity, right? Saying that these spindles can reform into bipolar spindles. And so we are currently doing cryo-ET of this double knockdown to really understand how these spindles now restore bipolarity, and how the spacing as well as actually the polarity of microtubules is changed in these spindles. And so just to sum this up, so this is the textbook cartoon. And so what we see is that there's actually, and I didn't put this here, so kinetical microtubules do not connect to spindle poles. There is a lot of antiparallel microtubules in these spindles. We know that kinesin 5 is required for these antiparallel population, not bipolarity. We can see that dynein does not at all affect microtubule polarity. And we can see that kinesin and dynein really can modify the lattice spacing and probably thus affect the assembly of spindles. And I hope I can also convince you that this textbook model is really incorrect, right? So that there is not only 3 classes of microtubules, or 2, kinetical microtubules and centrosomal microtubules. There's a lot of microtubules in spindles that are not connected to poles or chromosomes, and those microtubules are also not oriented all in the same direction. They're actually antiparallel, and so I think this textbook model could maybe be modified in the future. What we still don't know is how and where these antiparallel microtubules actually originate. So we do not know if they're maybe originate at the centrosome, so the main hub for microtubule nucleation, or if they're nucleated in the bulk, maybe by an augment-mediated mechanism. And we also don't know how the kinesin actually generates this population, whether it transports these microtubules, or whether it can just kind of catch little stubs of microtubules that are nucleated in the bulk and just orient them in a certain way. And so these are all questions we're trying to address now, and hopefully we will find an answer. And so with this, I'm at the end. And I want to thank, of course, Will Conway, who really did a lot of this electron cryomicroscopy, my lab, all our collaborators, the EM facility here at UVA, as well as all our funding sources. And I think maybe I stop sharing and maybe I can see actually someone here. Thank you.
Thank you, Stefanie, for that amazing talk. While we wait for any questions, I'll actually kick off with one. So with the dynamics with the whole spindle structure and all, have you considered looking at any sort of, or would you be considering looking at any, like in terms of disease context, like aneuploidy or cancer or something like that? Because I guess there'll be a lot of variation in that then.
Yeah, totally. So I think, I mean, there's always 2 things, right? One is to have a very clear understanding how a control spindle actually looks like,
Yeah.
right? And so I think that There's a lot of work maybe to be done too to really know how spindles look, and then it
Mhm.
would be easier to compare it to disease. But a lot of these proteins, for example, kinesin 5, right, and dynein, if they are affected, then spindles are doing very badly, and you do have increased aneuploidy,
Yeah, yeah.
right? So I think in at the end you can use it for this, but
Okay.
yeah, I think we still need a very clear
Yeah.
picture of how.
Yeah. And would you be doing it across different organisms as well, like something like Drosophila or something, or different
So
more?
yes, the plastic we've been doing, right? So I showed some mouse
Yeah,
stuff,
yeah, yeah, yeah.
and
Yeah, that's
we've done it in different cell types. Yeah, I mean, for the cryo, it would be amazing too. But if you've ever tried it, right, there's a lot of challenges.
it's
It's
yeah, yeah,
really
yeah.
not easy. And I mean, I didn't say, but it took us almost 2 years, okay, to get to some meaningful approach to know where are you in the spindle, right? And how do
Yeah,
you get to the
definitely.
cell? And
It's a lot of work.
it would be amazing to do it in a mouse. And I think long term, this is definitely something we
Yeah,
would want to do. But I also feel like you got to start easy, and then.
build up from there. Yeah, that's true. Um, we actually have a few questions from Boyan, if you will want to go ahead. I think,
Uh, Hi, Stephanie. Thanks for the great talk. This is Boyan from La Trobe. I was just wondering if the missing wedge artifacts was a big challenge for aligning the plastic zero sections. I did a bit of a zero section tomography before, but when aligning the zero sections, I had to sort of take a guess or do a bit of manual alignment because because of the missing wedge artifacts, the image, part of the image looked a bit of a smeared out or blurred. So I'm just wondering, was it a big challenge for you, or was there any new software or new algorithm to assist this process? Thanks.
Yeah, so actually it was not too difficult, but so I don't know how you did, but so when we take the tomograms, we actually do dual tilt, right? So we tilt first in this direction, and then we rotate the tomogram 90 degree, and then we tilt in this direction, and then we combine basically these A and B stacks, and we do this all in iMod actually. And so it's not too bad for the alignment because vesicles are very well visible, and then when we align sections in Z, we basically use a coarse grain model first, where we align vesicles on top of each other, and then we use the microtubule segmentation data to do a fine alignment. So maybe that helps it, but no, I think it's not too big of a problem. But again, I don't know if you also did dual tilt. Maybe that already makes a difference.
okay. Thanks for that, Stephanie. Our next question is from Georg.
Can you hear me?
Yeah, we can hear you, Georg. Go ahead.
Okay, cool. Cool. Thanks. Fantastic talk. Really enjoyed it. Yeah, so many questions actually. Yeah, it's amazing that you can actually see the different orientation of the tubules. Is that restricted just for the kinetochore microtubules, or do you see it in the periphery as well? That would be my first question. The other question is the cell still function if you lose the mix in the orientation of the microtubules? Do you still get spindle separation and all these kind of things?
Okay. So actually, I think so. Kinetochore microtubules have mainly uniform polarity. So it's the other microtubules that are antiparallel. So kinetochore
Yeah.
microtubules we see have their plus end at the kinetochores, but then the surrounding spindle microtubules are mostly mixed polarity.
Yeah.
And then are the spindles functional if that's a very good question. I guess not. So because so you lose the antiparallel microtubules if you treat with monostrol, and then those spindles are monopolar and they're certainly not functional. Yeah, I don't know. I mean, it's kind of, on the other hand, we don't know. I mean, if you inhibit the kinesin later in a spindle in anaphase, right? They are still undergoing anaphase, so then they don't seem to disturb. However, I don't know if polarity then is affected. right? But I think it's a really good question because, of course, now once you see those microtubules, you have to ask yourself, like, is there any functionality to them, right? And maybe they're just providing stability at the end, because if you have different polarity, cross-linkers may work better. Some motors can cross-link, so maybe it's just more stability. But yeah, this is something we're actually trying to understand now. What's the effect of those microtubules and what's their purpose, if you can say it like this.
And I mean, the other question was, is it something you only see in the spindle, or do you see it in the cell periphery as well? Is it something that could be more common, or you think it's specific for these for the spindle apparatus?
We haven't looked in the cell periphery, right?
Yeah.
So yeah, I actually don't know. So this is something we should definitely do, but we were so focused on getting the spindle, right? And because you kind of cut away everything else, you're kind of left with this small area. But yes, no, I don't know, but I think it would be interesting to see. And I mean, you can think of also looking in neurons, right? Where people have suggested that in axons, you have all these antiparallel microtubules, right? So you could totally look there. I think that could be very interesting.
Okay, great. We have a question from Jing Jian. If you would like to ask a question, go ahead, Jing Jian. Are you there or? I can't see the question actually.
There's one in the
Yeah, from Jing Jian.
chat, right?
Yeah. Stephanie, if you can see the question, I can't see the question actually. I'm sorry.
Yeah, I can read it. It says, okay, have you considered using methods such as polarized SIM to image the dynamics of spindle assembly process? I think there were a lot of work based on polarized microscope to observe spindle, but super resolution methods may give. So have we tried super resolution? And no, we have not. And I think maybe for a dumb reason, because when I started, super resolution was still very slow. And because we're interested in a highly dynamic process, we have just never really entertained it. I think these days, super resolution is much faster, and maybe it would be worth another try. But we have actually not done it. We've done mainly the three-dimensional large-scale reconstructions, and now we've moved into a higher resolution.
Okay. Okay. I don't think if there are any more questions, I don't think there are. So with that, we'll wrap up over here. Thank you so much for your time, Stephanie. I know you've it's late evening over there, and you're probably
Yeah, it's
trying to.
now it's very dark.
Oh yeah, that's dark. Our days just started. So, but thank you so much for your time. This was a beautiful talk, and we've all enjoyed it and learned so much from it. And good luck with your future work.
Thank you. Thank you for having me.
Thank you
And
so
have
much.
a nice rest of your
Thank you very much.
Thank you. All right. Bye-bye.
I mean, I have
Bye.
many more questions, but
You can still
you
go ahead, Georg, if you want.
Yeah.
oh, okay. It's difficult to AI. Is it okay to still talk or not? Okay.
I'm still here.
Can I show my pictures? Anyway, yeah. So you also had the you showed the graphs for the different phases.
Mhm.
So did you do the reconstruction for the full for the full spindle for all the different phases as well?
Yeah, so we did metaphase and anaphase and
Yeah.
mitosis, and then meiotic spindles we did actually early anaphase and late anaphase and metaphase and early metaphase. And in mouse, it's currently only metaphase because it's easy. So we do mouse oocytes, so it's easy to arrest them in metaphase and then take images there. Yeah.
Okay. How long does it roughly take to take such a full? I mean, it's a lot of work. I mean, if you
Yeah.
have to do the serial sector, the tomogram, double tomograms of each of these things. I mean, how long does it take to take a full set from the spindle apparatus?
Oh, well, that depends. If a spindle is small, it can be done in 3 months. But I think often the problem is that you need a really good data set, you know, where you really collect every single sections. We can't have any
Yeah.
gaps. Yeah, I think long term, you know, it would be better if one just takes smaller areas
Yeah.
and use this. You know, you don't always have to do a full reconstruction. Sometimes just the area around chromosomes or a smaller half spindle may actually be sufficient. But yeah, we're doing a mouse spindle, and that's just crazy many tomograms. And then, you know, it's like 50. And so you have to also have 50 serial section cut tomograms. So I think there's a lot of room for, you know, sample loss or whatever can go wrong.
Yeah, very, very cool. Very, very impressive work. And for the cryotomograms, some of the images you showed that was from the waffle, I think, in the lower right. So there was some thing that was waffle milling and trench cutting, and so did you do a mixture, or did Will do the mixture of different techniques?
We tried the waffle, but it was just too complicated. So you end up with, it's really hard to find your cell in the waffle because you end up with a sheet of ice. And so we figured that, because realistically, waffle would be better for freezing, right? I mean, we can all agree that plant freezing is a compromise here.
Yeah.
But then targeting just became so much more complicated.
Okay. Yeah.
Yeah.
I see. Okay. Oh, well, fantastic work. Thanks so much for the talk.
Yeah, thank
I
you.
really enjoyed it. And yeah, thanks
Great.
for, yeah.
Thank
Okay,
you.
cool.
And thanks, Stephanie. Thank you so much.
Thank you.
Thanks, Natasha.
All right. See you.
Thank you.
Thank you.
Bye-bye.
Bye.
Bye-bye.
Bye.